Good evening, everyone. This is Harshvardhan on behalf of Boltzmann Labs team, and I welcome you all for this webinar, which is Hack the BoltzHack. Firstly, I would like to congratulate you all for registering for the hackathon event, which is on November 26th to 28th. And this two-day webinar will give you an insights on how you can work on Boltzmann AI suite and in what ways you can use the suite and what kind of problems you can solve. And I welcome now Catherine. She is Cheminformatics Scientist who has been working with Boltzmann since 3 years, and she'll be taking you over about the AI suite that is developed by Boltzmann Labs, and also she'll be talking about small molecules design. Catherine, please go ahead.
Okay. Am I audible?
Yes, Catherine.
Okay. Good evening, everyone. I'm Catherine, and like Harsha said, I'm the subject matter expert for small molecule design here in Boltzmann Labs. So for today, I will initially walk you through a few of what small molecules mean and what are the processes in small molecules, how you can kind of find its alternatives in silico, why in silico is required, and just kind of give you a preview on the functionalities that is available in Boltzmann suite, so that you can come up with your own strategy to design and to design, discover, and do your thing in the Boltzmann suite for the hackathon. So I'll just AI in small molecule discovery. So before I get into it, for those of you who are not so familiar with this particular field, what is a small molecule? Now, all of these are small molecules. So not all small molecules would look similar, but you get the general picture. Small molecules are nothing but molecules that have that are smaller in the stratum of all the molecules that are out there. So the spectrum of all the molecules that are out there. Considerably, we would say molecular weight of around 1,000 to 1,500 Daltons, and everything that is lesser than that would be considered under the designation of a small molecule. Now, we might be familiar with different types of small molecules. For example, right now, what we have here, the initial one is an antibiotic. It's penicillin, and one of the first antibiotics, or the first antibiotics to be discovered. And then you have something called mesotrione. Now, this is a herbicide. And then you have this particular chemical entity, which is nothing but salicylic acid. It's a very common cosmetic agents that are used for a bunch of cosmetic purposes. Now, in all these fields, you have small molecules, playing a role. So, if small molecules are so widely available in our day-to-day activities, how is small molecules, being discovered or designed? So this is a general workflow on how small molecule drug discovery works. So drug can be, in a general term, I'm using it as a pharmaceutical drug. Obviously, the processes would change a little bit depending, or at least the final processes of preclinical studies and clinical trials would change depending on the industry that you're working with. For example, cosmetic and agrochem may not require the kind of strict restrictions and safety principles that you would have with pharmaceuticals, but this is a general discovery flow that happens with small molecules. So you have an initial, the initial step of target identification, wherein which you're trying to identify the protein target for which you're designing or discovering a drug. Now, if you're looking into the in silico part of it, there are multi-omics approaches that are available, or if you have literature reviews that are pointing you towards a target of interest for a particular disease, or in other case, you have a target that you're working on. any of that, you have your target. Now, what is the next step? So if you don't have a molecule at all, there are no previous molecules that are designed for this particular target, you're going to go around with something called hit identification, where you're trying to identify a molecule that can bind and inhibit the identified target. Now, you're not trying to look for a molecule that has very strong potency or a very strong binding. You're looking for something that has moderate binding. So, for example, if any of you are from, in vitro background, high throughput screening is a very common method wherein which you give around 10,000, 50,000 so ligands, and then you try to see how their potency is with respect to the particular target, or how well they are binding to the target. So that is your hit identification stage. You're just trying to identify a molecule that have moderate binding or a moderate activity. After that, you're trying to optimize the molecule. So in this case, in the second stage, where you're trying to optimize the hit to have a better binding or a better inhibition, you call them something like leads. Now, in this case, you're trying to optimize the molecule in the sense you're trying to keep the majority of the structure the same as that of the hit, and you're trying to change certain parts of the molecule so that it would have better binding or it would have better inhibition and so on. So usually in case of previous in vitro studies, chemists would try to study the binding pocket of the particular target and identify certain pockets, whether it's hydrophobic, what kind of binding works. So based on all of that, they try to kind of change and optimize the structure of the hit molecule so that they can bind better to their protein target of interest. Now you have the third stage, which is your lead optimization stage, wherein which you are trying to optimize the molecule, not just for the binding and inhibition. Obviously, that particular molecule has to be safe for human consumption, or even if it's in the case of a herbicide, even if a human comes in contact with that herbicide, it should not be lethal to us. In case of like agrochem based molecules, it should not be lethal to the crops of interest as well. So based on the safety profile on the entities that you're trying to prevent its activity from, we have to see whether it's safe, whether it's non-toxic. So in this particular stage, again, you're trying to optimize the structure to kind of be safe with respect to what you're trying to protect, whether that is human physiology or whether that is a crop, depending on whichever industry you're working with. And then you have your preclinical studies, so wherein which you're testing the molecule on some animals, whether that be mouse, rat, or other preclinical studies. And then you're going on and testing these molecules in humans, which is where your clinical trials are, and you have stages of those clinical trials, which is assessing the efficacy of the drug and also the safety of the drug. So these are the major stages within drug discovery, and these are major stages that has been adopted for drug discovery, not just in silico, but this has been the traditional route of drug discovery that has been happening over ages. And almost all of these stages, which is hit identification and hit leads and lead optimization, usually happens usually in a lab setting. Now, these are the amount of money and time that you actually spend on these drug discovery stages. So you have your target, you have your target to hit to lead, lead optimization, preclinical, phase 1, phase 2, phase 3. And you can see that it takes around 1.8 billion just in development, and that is just the metrics from 2010. And these are the, cost per millions at each different stages. Now, I'll just show you this metrics. Now, these metrics are nothing but the failure rates. So you're expecting at least the 37% failure rate. So you're going to take a molecule from lead optimization to preclinical, where you're trying to test the safety of that molecule in a living organism. You can expect at least 37% of failure. So if you're taking 100 molecules, 37 molecules are for sure going to fail. And then you're going to take these molecules again into clinical trials, where you have a very high failure rate of 90%. the chances of your molecule, even though it took up these many millions of resources and these many years, which is around, say, you have around 3 to 4 years in just in vitro studies, you're going to face a 37% failure rate in animal studies and a 90% failure rate in humans. So what we are trying to do here is we are trying to optimize, or we are trying to reduce both the time that you're going to spend on these molecules and the cost that you spend on these molecules. So now there has been number of methods where scientists have tried to optimize it. So for a long time, we had something called CAD, which is computer-aided drug discovery. Now, I guess some of you might know, we have molecular docking, molecular dynamic simulations. These are very old methods that has been employed in drug discovery, wherein which you're trying to study the binding of the molecule before the molecule, are being synthesized or tested in lab. Molecular dynamic simulation, you're trying to study the stability of the molecule. So different methods you have been used to study how the molecule binds so that we can reduce the failure rate. Now, that reduction in the failure rate and the time that is taking is, it has been reduced to a certain extent. Now comes into picture AI. As you can see, the time that has been taken by or a drug designing or drug testing using AI is way lesser than what you could see in CAD or what you can expect in an in vitro setting. So a molecule design, you can easily do it in 1 to 2 weeks. Considering you have your inputs and your hypothesis straight, you can do molecule design in just 1 to 2 weeks. And you can optimize the said molecules for ADME properties or any toxicity endpoint that you have, again in 1 to 2 weeks. This is giving you around 2x reduction in time, 3x success, or a 3x increase in success rate in clinical trials. You are looking into chemical spaces which maybe we haven't tried to look into before, and there is a significant reduction in cost. So this is how AI kind of binds into how a small molecule design or a discovery works, what it actually does. Now, keeping into account the discovery flow of a small molecule, I would like to kind of present to you guys the Boltzmann platform and the kind of functionalities that we present. Can you all see the platform?
Yes, Catherine, it's visible.
Okay, so this is where you're going to go into the platform, or the platform that will be made available to you when you guys join the hackathon. So you have a few, on the sidebar, you have a few functionalities, which is your dashboard, the projects that you're going to build, the workflows, the AI agents, and so on, which you're allowed to look into in your own leisure. And today I'll be going through the small molecule suite. Now, under the small molecule suite, you will have a bunch of subdivisions. I'll not go through each one of them and all the functionality that it presents, but I'll kind of try to correlate with the drug discovery flow that I had mentioned and how it all ties up together. So initially I mentioned we have something called target identification. Now, target identification is the only part of small molecule drug discovery that has not been added to the platform as of now. And the second part of it is hit generation or hit identification. Now, for hit identification, we have two separate modules that is working. One is generation of hits. So in the generation of hits part, what we are trying to do is we are trying to employ AI-based methods to generate new hit molecules. Now, the inputs to generate new hit molecules varies with the tool that you would be selecting. As you can see, you have something called structure-based generation over here. Now, structure-based generation will help you generate new molecules based on the binding pocket of your protein. So if you go to your structure-based generator, only thing you have to add is the binding pocket. Specify the number of shape. What shapes exactly mean would be there in the user manual, but for your reference, shapes just means how much or different regions of the binding pocket should the AI kind of sample, and how many molecules you want out of them. And if you want to predict any of the toxicity endpoints, you can predict it by choosing them, and you can generate a set of molecules. Now, I'll just show you real fast. So these are some of the molecules that has been generated from SBG, and only by giving the binding pocket of the protein that I've of interest. Now that is one particular module. There are a bunch of other modules. For example, something you might be familiar with is pharmacophore-based generation. If you have a pharmacophore, whether that be a ligand-based pharmacophore or a receptor-based pharmacophore, you can go over and give that pharmacophore as the input and generate molecules. You have your random generation, wherein which you can randomly generate molecule, and you can specify you can build models and you can specify the ranges within which the molecules has to be, they have to be within. So all of these modules might sound a bit too complex at the moment, but during the hackathon, we'll give you a more detailed explanation of all of them. So this molecule generation module basically helps you to generate molecules de novo just by using different kind of inputs and whatever kind of inputs you have at hand, whether that be hit molecules that you had previously, so it's like a hit expansion study, or whether that be the binding pocket or the pharmacophore or a single molecule that is a hit molecule. You can use that as well to train your model and generate new molecules. And the other method is virtual screening. Now, say you don't want to generate new hit molecules, you want to discover them. So discover in the sense that there are a bunch of molecules that are out there, for example, like repurposing, like drug repurposing. You have a library of molecule. Say you downloaded a set of molecule from the Campbell database, or you downloaded from Zinc database, or you have a set of molecules that are known to be approved molecules, approved small molecules. You can use different types of screening methods that are available in the Boltzmann suite to screen in these molecules. Now, you can screen the molecules based on their similarity. So you know that there is a molecule that is a drug molecule for a target. You want new molecules, you can screen them using their similarity. You can screen them based on the pharmacophore, substructure, if they have a particular substructure at hand, QSPR screening, which is just looking into different kinds of, models. So these are basically assessing the admit properties or the admit profile of your molecule. You can choose any of these properties and it will filter your final set of molecules to that. And you have novelty toxicity. So these are all screening methods that you can use to identify hits. Now, the virtual screening flow is not just it is not restricted to finding the hits. You can, like I mentioned, you can use it for ADMET-based screening, or you can use it for toxicity-based screening, or you can use it for novelty-based screening. This module is just going to check whether your molecule is novel or not. So these are just few screening modules that are available. And so that was the first two stages, right? You identified your hits, which is basically you're trying to identify molecules that are initially binding to your target. And then you can use a bunch of these. So these are probably tools that are more familiar to you. You have molecular docking, binding site prediction, binding affinity prediction. All of these modules will also be available. So say you identified a hit, or you identified 100, 200 hits from screening or generation, you can use your molecular docking module to dock these molecules, and you can see, you just have to upload your ligand file and your PDB file. So you can upload how many of ligands you want. You can upload, I would say a max of 500, anything less than 500, you can add, and you just have to give the smiles of the said molecule. You don't have to upload the 3D structure as well. You have your ligand file, you have a PDB file, just mention the center, the radius, and then the exhaustiveness. And basically what we are running on is Bina. We just made the output and input very easy for the user to kind of work on. Or if you want, you can do a blind docking. In this case, we are using a DL-based method called diffdock. You just have to upload your protein PDB, again, your ligand file. You can either upload or you can even draw. So you have your sketcher over here. You can draw your molecule that will be added as mine. So either way, you can do it. So once you have your hits, you can use all these other modules like lead generation, molecular optimization, or fragment-based optimization to optimize it. The major difference between your hit generation modules and your optimization modules is that, now if you remember, I mentioned that what happens exactly in the hit optimization is lead optimization is you're trying to optimize the structures to have better admit profile so that they should be safer. So imagine you generated a hit and the hit works really well for inhibiting the target, and then you found that your hit in a preclinical setting or a clinical setting is not as bioavailable. That means it doesn't go into the stream as well. Okay, maybe simpler. You found that the molecule inhibits your target very well, but it's carcinogenic. Now you want to mitigate that toxicity. So only thing you have to do is, again, go to the Boltzmann platform, go right here, you have molecule optimization. This is the lead generation, molecular optimization. I'm just going to pick one. And then say I have a molecule at hand. I found that this is the molecule that is working very well. This is the molecule that is working really good as a hit. And I want to optimize it so that it doesn't have any carcinogenic profile. So over here, if you have all of these ADME properties that are being mentioned, I'm going down to the toxicity. I'm choosing carcinogenicity. And then I'm saying I want it to be classified as non-carcinogenic. So in this case, I want it to be classified as 0. So I would give it a range from 0 to 0.5. And then only thing I have to do is mention how many number of molecules I want, whether that's 1,000 or 10,000 or so on. If I have any QSAM models built, I can choose that as well. And then I can go and click here, submit. So from the structure of the same molecule, it will try to optimize it. So it won't change the structure completely. It will optimize certain regions of the structure, looking for molecules that are non-carcinogenic in a similar space. And you don't just have to restrict yourself to one model. You can choose any model right here. You can choose blood-brain barrier, whether you want it to be non-permeable or permeable, whether you want lipophilicity in a particular range, half-life, clearance, metabolism. So you can start with any molecule structure and optimize any number of ADME parameters that you can. Sometimes you might not find molecules that are serving all the parameters. For example, you give say 10 or 20 ADME properties. You might not find molecule that satisfy all of them in all the given ranges, but you will find at least molecules that are satisfying 50% or 75% of the properties. So that are a few functionalities that are available in Polychem or small molecule discovery suite. So I will quickly walk you through a case study that we did. So it might be a bit simple for you. So this is a full-on case study wherein which we have generated hits, and then we have generated leads, and then we have optimized that leads. So what we did was we built a QSAM model first, and then we generated hits. We went to hit to leads and lead optimization. So all of these modules are available. In case once you've joined the hackathon, I will provide the same or this particular case study to you for you to kind of get a better clarification on how we did it. So what I would like to kind of focus on is how the molecule properties have improved. So these are your reference molecules, right? And these are the molecules that we generated. Now, these molecules are structurally very different from that of your reference molecule. But even then, in the initial stages, these are the molecules, these are the predicted inhibitory activity, and these are the values of how strong they're binding. And then this is our hit generation, this is our lead generation, and then we have these values. And in the third stage, which is our lead optimization. And these are the values that we got. So as you can see, there is an iterative increase in the predicted activity as well as binding affinity. Binding affinity, so basically this is essentially binding affinity, but then it's basically your docking score that we got from winner. So there is an improvement in the docking score, almost matching that of the reference molecules. And then you have your predicted activity, which is found again to match that of the reference molecule. So these are certain profiles. These are our MDS profile. We found that these molecules are synthetically accessible in terms of the complexity of how the molecule is. We have the binding pocket is similar, and they're all stable during MDS, which means that they could be potential molecules that we can move forward with. So this is a very small case study that we have done in the Boltzmann platform. During the hackathon, there is no need to do a case study that goes all the way from hit generation to lead generation and lead optimization. If you have a smaller case study that you're interested in, which is say a virtual screening to optimization, a smallest case study, that is also possible. But all of these modules are present here. You have your QSAR module also present, so you can build whether that is machine learning models or deep learning models. You can choose either descriptors or molecular fingerprints, so any of these you can choose. So all of these modules are present to do an end-to-end small molecule drug discovery flow. Though how you want to design your flow is completely up to you. So these are just basic functionalities that is present in small molecule design. Does anybody have any question? Just a minute. Which chemical database are you using, Sharia? Could you kind of expand on that chemical database in what particular part? I mean, in which particular section did you? I mean, which particular section were you referring to with respect to chemical database?
Sharia, you can unmute and ask your query in detail.
Okay, in molecule screening. Okay, so basically we're not giving any library as such, but you can bring your own library. It depends. So for example, you can go and download approved molecules, say from PubChem or FDA site, you can download approved molecules, or Chembl, you can download whatever molecules are known for a particular target. You can do that, but we are not providing any database as such. But if you guys have any doubts during hackathon, I can suggest some databases that you can probably look into.
Yes, Sharia, like in the hackathon, you will have to use this platform and make the case study or whatever the problem that you are going to solve. Like we'll be giving you the access on the day one of the hackathon. Is there any other queries by any other participants? You can unmute yourself and ask either me or Catherine, or you can just simply drop in the chat box. And what would be the evaluation criteria? Shaurya, yes, evaluation criteria will also be announced on the day of hackathon. Like day one morning session will be the kickoff session where you'll be given the access and where you'll be given the rules that are to be followed and what are the evaluation criteria. Can we participate in both the tracks simultaneously? Yes, you can participate in both the tracks simultaneously if you are able to manage it. You're always welcome for that. Any more queries? Will we be assigned any particular problem to solve? No, we are just giving the themes, that is protein engineering and small molecule design. You can come up with some innovative problem and its solution. Like you'll have to come up with some innovation. That is the main point of hackathon. And Anil, will we get recording for session? Yes, you can drop us an email. The email ID is contact@boltzmann.co. I'll be sharing you the recording. You can just email us. Okay, before I take up more queries, I would just like to thank Catherine for this wonderful presentation, for clarifying what's the hackathon going to be like. So just to summarize what Catherine has told, like she has discussed about the small molecule basics and what is the drug discovery process, that is target identification, hit to lead generation, lead optimization, and also the clinical pipeline that she has discussed. Also, she has addressed the industry challenges such as developmental costs and high failure rates, and also the time investment which are caused due to the traditional methods. And that is why we have bring this AI suite from Boltzmann Labs, which will reduce molecule design time and 2 times reduction and 3 times increase in the clinical success rates, and also significant cost reduction compared to the traditional methods. And also you'll be getting access to previously unexplored chemical spaces. Shaurya, to address your query, like how our results will be used by Boltzmann. So whatever the problem or the innovative solution that you're coming up with, that is like solely will be yours. But if you want to do any collaboration with us, we are always welcoming students for that. Yes, Anil, you have to come up with your own innovative